Hello everyone, welcome to yet another session with online biology tutorials. We are here to discuss the second part of the Human Genome Project. In the first video of Human Genome Project, we have already discussed why it was called as a mega project, what were the goals for this, what were the methodologies adopted. Now we will move ahead quickly and we will see what were the steps to sequence the human genome. Okay, so the first step was the isolation of DNA. So DNA was isolated, and if after isolation the quantity of it was less, then its quantity was increased by PCR. So second step was done only if the quantity of DNA was less. So PCR stands for polymerase chain reaction. This is a process that we will study later. In this process, the DNA quantity can be increased. Okay. Now, after DNA was isolated and if its quantity was less, polymerase chain reaction was done, and then the DNA was treated with restriction endonucleases. Restriction endonucleases are the enzymes that will break the DNA in between. The activity of these enzymes will be more clearer to you once we will come to the chapter of biotech, because there it is in detail. So once they are treated with restriction endonucleases, the DNA after this was separated. Okay, once it was separated, it was introduced in the host for expressions. So next point will become introduced in the host for expression. Every step will be much clearer to you because it will be told in detail in the biotechnology chapter. Here you must know that two hosts were required. One was bacterial artificial chromosome and another was yeast artificial chromosome, right? So this is bacterial artificial chromosome and another was yeast artificial chromosome. So two hosts were taken. One was prokaryotic in origin and another was eukaryotic in origin. Once the gene expression was done in these hosts, then their product was collected and it was sequenced by Sanger's method. After sequencing, overlapping was done to find out what would be the sequence of the nucleotide. Okay, so this is the whole procedure that was adopted, and by this sequencing was done. You need to learn each step, and the next point which we are coming on to is the salient features of Human Genome Project. Okay, salient features will be the one that will tell us what would be the findings of this project. Okay, so overall it was found out that 3.3 into 10 to the power 9 billion base pairs are there. So BP means base pairs in human genome. Okay, and it was estimated that human being consist of somewhere around 20,000 to 25,000 genes. Okay, every point which we'll be writing in the salient features, they are important, like how much will be the base pairs they have found, what would be the total number of genes, and it was also found out that 50% of the function of genetic material is unknown. means 50% of the genes, they were not knowing, the people who dealt with it, they were not knowing that 50% of the genes, they were unestimated, means their function was totally unknown. What are they doing? Okay, so 50% function of the genetic material or genes was unknown. It was found out that only 2% genes, they help in gene expression. Okay, so only these many percent of genes, they help in the gene expression. It was also deduced that we resemble to each other in 99.9%. That means you resemble to me 99.9%, and so as I resemble to everyone on this earth. We are dissimilar to each other just by 0.1%. Okay, so you can frame the sentences that we are similar to each other in the genetic material, in the number of genes by 99.9%, and we just differ with 0.1%. Okay, so why this only has the difference? Because this includes the repetitive DNA. Okay, so we are dissimilar to each other because we have dissimilar repetitive DNA in our body, right? So you can write about this repetitive DNA, which counts for 0.1%. It exhibits polymorphism. Okay. So polymorphism means, poly means many, morphology means the look. Okay, so because the repetitive sequences are different in your body to mine, that is why it was said that polymorphism is there in the human body, right? Another thing which they found out is that the average length of gene was found out to be 3000 base pairs, and the longest gene that they discovered was the dystrophin gene, and it contained 2.4 million bases. This was also the finding of Human Genome Project, that if you take out a gene, its average length will be 3000 base pairs. But if you talk about the longest gene, which is dystrophin, it had 2.4 million bases, right? Another thing that was found out was the chromosome number Y contained the least number of genes, and the number of genes that were there on chromosome number Y were 231, whereas chromosome number 1 was the longest one in terms of the genes, because it had maximum number of genes and the number of genes on it were 2968. So the numbers of genes which are written for chromosome number Y and chromosome number 1, this has to be learned. This is important. So salient features, let's sum up once again. What were these? So number of base pairs in the human genome, they were 3.3 into 10 to the power 9. The number of genes that were found out, they were from 20,000 to 25,000. Means each human being has 20 to 25,000 genes. 50% of the genes, their function was not at all known. Only 2% of the genes, they help in the gene expression. We are similar to each other in 99.9%, and we just vary with 0.1% because that is the repetitive DNA that varies from me to you. That is why we call it as polymorphism. Maximum number of genes were there on chromosome number 1, and least number of genes were there on chromosome number Y. If it is asked to you which is the longest gene, means they are asking you in terms of the base pairs. So longest gene will be the one that will contain maximum base pairs, and it is dystrophin with 2.4 million base pairs. Whereas the chromosome with maximum genes is chromosome number 1. The chromosome number Y contains least number of genes. So these were the salient features of this project. Now, not only human genome was sequenced, later other organisms like Drosophila melanogaster was also sequenced, right? So if it is asked to you what is the other organism that has been sequenced, so answer is Drosophila melanogaster. E. coli has also been sequenced. So these are the scientific names. Whenever you are writing them, please do underline. C. elegans is also an example of it. So these organisms, they were sequenced with an intention that if we want to do any genetic study, we can apply on them and we can see their effects. Okay, so salient features of Human Genome Project are very, very important. I would advise that if a subjective paper will happen, then you should not leave any feature. Every feature is important. Last but not the least, we are heading towards the application of Human Genome Project. Now, if the entire sequencing of the genome is done, how it could be helpful? Many diseases in the human beings, they are happening, and if we could really find out that which gene is affected in that particular disease, we can work out for the medicines and we can give a cure. There is one disease which is known as diabetes, and diabetes is a disorder of the pancreas because pancreas is the only heterocrine gland in our body. If I talk about its endocrine function, it produces insulin, right? So if insulin is not at all produced, then it leads to a disease called diabetes. Some people, they have varying degrees of diabetes. Some produce little insulin because their pancreas function a little, whereas some, they don't produce at all insulin. So the people who are not able to produce insulin naturally in their body, they take it externally with the help of intravenous injection. Now, insulin is a product which was a biotechnological application. So the sequencing of insulin protein, which is actually a hormone, it is a proteinous hormone, its sequencing was done, and that's why it was artificially synthesized. So likewise, we can estimate for many other diseases and then we can give a cure to them. So the major application of Human Genome Project is in treating the diseases that are affecting the genome of an individual. A therapy can be given to it. Okay, so in Human Genome Project, overall we discussed which were the agencies that initiated this project, what were the goals, why it was known as the mega project, what were the methodologies, what were the steps that were used to do the sequencing, salient features, and in the end we discuss the application. So both the videos on Human Genome Project are important and sufficient for your studies. Stay tuned for more videos. Thank you.